so many prizes, I really don't have time to list them all. But one thing that really struck me in this bio that they sent preparing for this was he mentioned that he's written over 1,500 articles, cited over 382,000 times, and his H index of 307 is the highest of any engineer in history, and the second highest overall. And I was explaining how impressive this is to someone, and I realized that a lot of people who are not active in academia or scientific publishing, like myself, have no idea what an H index is. So I thought I would explain it. So the H index is based on the number of documents, the number of citations. It is the number of time, it is the point at which the number of, you have written X number of papers that have each been cited at least X number of times. And we usually, this is calculated by creating a histogram of, for each article, how many times has it been cited, and then just simply drawing a line with slope equals 1, and where do they cross. And at the gold star, you can see this is, it has so many citations that we needed to do a, I thought I'd do a little bit of an enlargement there. So you can see that that's, that's crossing right at about 307, maybe a little lower. This is a different, slightly different data set. But yes, that's what the H index is. This means that his work is, it's not just a lot of articles, it's a lot of articles that people have read, have built on, and have published further articles building on that and moving science forward. Again, we're all standing on the shoulders of giants. Nice colorful look at just the areas in which he has published. Very multidisciplinary. The sort of red color scheme is medicine, pharmacy, health, veterinary sciences. The blue is more chemical engineering, material science engineering. The green tones are biochemistry, biology. So you can see this in a very nice, this is a very colorful way to show that his work spans the gap from medicine to engineering and everything in between. And that is, that's really all I have to say. So I know you're all ready to hear from Bob Lander.
Well, thank you so much. It's a pleasure to be here. I'm going to start with me being at MIT. So I got my degree, my doctorate, in 1974 in chemical engineering, and I'm going to start from there and go to today. But so anyhow, when I got my degree, it was 1974, like I mentioned, and there was this gas shortage, and so what that meant, if you were in Boston, is that you actually had to wait in line for 2 hours to get your tank filled, for those of you that are old enough. But what that meant for chemical engineers is you got a lot of job offers. and so almost all my friends, they went to work for the oil companies. So I thought I'd do what everybody else did. I would, they'd keep coming to campus, so I actually got 20 job offers, 4 from Exxon alone. Believe me, I wasn't that good, but they had so many openings, and I still remember going to one of the Exxons, and one of the guys there said, "Gee, if you could just increase the yield of this one chemical by 0.1%, he said, "that would be just fantastic. It'd be worth billions of dollars." And I remember flying back to Boston that night thinking to myself that I really didn't want to spend my life doing that. and it made me think what I did want to do. So when I was supposed to be doing my thesis, I actually helped got a school called the Group School, started with some other people to help working class students in Cambridge. And I got very involved in creating new chemistry curricula. One day I saw an ad to be assistant professor at City College of New York to do just that. So I wrote them a letter applying for a job, but they didn't write me back. But I liked the idea, so I found all the ads I could to do something like that. I found about 40 different places, and I wrote to all of them, and actually none of them wrote me back. So then but it made me think what I did want to do, and I thought, well, I'd like to use my chemical engineering education to help people, so I thought about medicine. So I wrote to a lot of hospitals and medical schools. They didn't write back either. one day I walked into my graduate lab, and one of the older scientists said to me, he said, Bob, he said, there's this surgeon in Boston named Judah Folkman, and he said sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. Anyhow, Dr. Folkman had an interesting idea, which is shown on the next slide. And if you look at this, it's, they didn't give me a pointer, so I'm just going to explain things. But basically, the whole idea is that he theorized that blood vessels were very important to the tumor growing, enabling it to get nutrition and get bigger and bigger, as you can see on this slide. But even more critically, he said the reason that happened is the tumors created a diffusible substance called TAF, or tumor angiogenesis factor. You see that there. And it's that chemical signal that causes the blood vessels to grow and feed the tumor and make it really big. And ultimately on the top, of course, the tumor can spread through those blood vessels. That's metastasis that often kills. So his theory was if you could stop blood vessels, that might be a whole new way to think about treating cancer. Now, I knew nothing about biology. I still probably don't. But he said, well, Bob, I'd like you to come and actually prove that I'm right and isolate the very first substances that could stop blood vessels from growing. And I thought, boy, this is fascinating, and if we could do that, that would be a big deal. One of the biggest things, though, when you try to solve a problem like this, is having what's called a bioassay, a way to study something unknown like this. So none existed. So one of my other jobs was to develop one. And what we thought about is shown on the next slide, which is that there are certain places in the body and also certain types of chip membranes, correlative membranes, but that don't have blood vessels, or very many. What if we put a tumor there and put next to it a tiny little particle, like a micro or nanoparticle, that could deliver anything we isolated to see if it could stop the blood vessels from growing, that you could really visualize it, maybe even quantitate it. So that's what I was going to try to do. Now, Dr. Folkman was on the board of the one company in the world, ALSA at that time, that had done any work at all on drug delivery. And he asked some of the top chemists, they had great chemists there, he said, could they help us? But they all said, well, trying to deliver a large molecule from a tiny particle is impossible. It's like any of us walking through a wall, just too big. And so what they said is that you could never get this to work. But if we were going to solve the blood vessel problem, we had to solve this problem. So I spent 2 years working in the laboratory. I actually found over 200 different ways to get this to not work. But eventually, I was able to make tiny little microparticles or nanoparticles. You see a picture of one here on the left, and cut in half on the right. And what we showed in the journal Nature in 1976 is this one approach enabled you for the first time to deliver large molecules from tiny particles. It was also the very first paper that showed that you could actually use an approach like this to deliver nucleic acids. Like nucleic acids are DNA and RNA, and I'm going to come to that later. I want to go back to 3 months before this paper came out in Nature in 1976. So now I'm less than 2 years out of MIT, and I got asked to give a talk in Midland, Michigan, which is a big materials center. That's where Dow and Dow Corning are. So I had to give this 20-minute talk. I'd never given a big talk before. I practiced this for weeks into a tape recorder. And finally the day came, and I gave the talk. I was 27 years old. And at the end of the talk, I thought I did all right. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought all these older chemists and engineers, being nice people, would want to encourage me, this young guy. When I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, we don't believe a thing you just said. And they said that, in fact, that these molecules are just too big to diffuse. Again, that's what you heard before. But also that the solvents we were using would destroy anything we put in. And things went downhill from there. I tried to get research grants, mostly from the government. My first 9 were rejected. The government, NIH, they'd say, well, Dr. Langer's an engineer. He knows nothing about biology, and even less about oncology. I thought I knew enough about math to know that was hard to do. But at any rate, I didn't do very well in that. And then I tried, you know, I loved being a postdoc, but a lot of my friends said it's probably not good to do that forever, so I applied for faculty positions. And of course I applied to chemical engineering departments, and no chemical engineering department in the country would hire me. They all said this bio work that I was doing made no sense for chemical engineers. I should be doing oil or something like that. So I didn't get any jobs there. But Dr. Folkman introduced me to Nevin Scrimshaw, and Nevin Scrimshaw was head of the nutrition department. And he was kind of a benevolent dictator kind of department head, for any of you that knew him. He liked me, so he offered me a job as assistant professor, but he didn't bother to ask anybody else in the department what they thought. which might have been okay, except for one thing, which is the year after I joined the department, he left. So a lot of the faculty decided to give me advice, and they said, well, I should leave too. This is a direct quote from Mike Marletta, who was one of my colleagues at MIT in 1979. And here he's saying, well, one evening he went to a faculty dinner at a Chinese restaurant with me and some MIT professors. He said one of them stopped quizzing us while smoking a cigar. He said when he heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. Well, I'm still here, but it wasn't so pleasant. At any rate, we had developed these approaches for drug delivery, and also I've been isolating these substances to stop blood vessels from it. Well, I want to show you what happened. Starting in 2004, 28 years after this paper, and using our techniques, Avastin, the very first of these blood vessel inhibitors, was approved by FDA. That is probably the number 2 or 3 best-selling biotech drug in history, and many, many others. And not only that, for the first time, drugs that could stop blood vessel growth in the back of the eye, like Eylea and Lucentis, which would become treatments for macular degeneration. Lucentis would also get approved by FDA, and these are really the only treatments today to stop macular degeneration or diabetic retinopathy, pharmacologic ones. So I'm going to go back in time, and you can even see 4 of these were approved in this past year, 2022. So going back in time to the 1970s, Dr. Folkman and I thought we would do something unusual at Children's Hospital, and that was file for a patent. That wouldn't be so unusual today, but in the 1970s, Children's Hospital had no patents. So we filed for one. 5 years in a row, it got rejected. And I remember the head of the tech transfer office there, Al Brazzini, came to see me one day. He said, Bob, you have to stop. You're wasting so much money. This guy will never allow the patent. And he said, you should just quit. But I don't like to quit. So I started to think, how could we convince the patent examiner to allow this patent? Well, legally, of course. And science really wasn't working. So I told you earlier that, so she deserves a medal. Thank you so much. So, yeah, so at any rate, so what I did is I did what's called the science citation search. Can people hear me okay? Yeah. So I started to look at what, as I told you, people said this was impossible. Well, did anybody ever write it down? So I did the search, and I found an interesting paper. I actually found a number of them. I don't think this changes the slides. So this is from 5 of the top material scientists in the world. They're just describing the field. And I have no idea this was written, but I'll just read you a little. They said generally the agent to be released is a relatively small molecule. One wouldn't expect that large molecules to be released like this because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, so I thought surprising, that's a really good word for patent examiner, to clearly demonstrate the opposite. So So I showed this to the people at the hospital. They showed it to the examiner, and he said, I'll tell you what, I will allow this patent if Dr. Langer get written affidavits from each of these 5 people that they really wrote them. So I wrote them, and they were all nice enough to write me back, even though I was assistant professor, that they really wrote it. And we got this very, very broad patent. On the next slide. You know, I got involved in starting companies, licensing things to companies, and today, because of that, many, many products are based on this are approved by FDA. Most of them are medical. In fact, some of the most common ways of treating opioid addiction, like Vivitrol, and Lupron Depot, very common way of treating advanced prostate cancer. Maybe that'll be better. At any rate, and even it's made an impact in the whole area of agriculture. Agriculture, that's on the bottom right. That's a $250 billion industry, and this enables you to control reproduction, even create, save fish species that are going to extinction. So at any rate, it ended up, these are used by hundreds of millions of people every year. Also, one of the things I noticed when we started doing started doing this is when we made particles smaller. Does that echo? Do people hear an echo? Anyhow, when people tried to make things smaller, what would happen is they'd stick together more macrophages, cells that could eat them. So we came up with this coating, polyethylene glycol, which is something that's in Miralax. It absorbs a lot of water. But if any of you have bathroom issues, Miralax is all polyethylene glycol. And only Vargazad in our lab it went further and put targeting molecules on these, so you could actually target them at particular cells in some cases. I sometimes don't explain this that well, so I wanted to show you a video that Nova, the TV show, did on our lab about this.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered. A key that will only fit the lock of cancer cells.
I should tell you that a lot of the clinicians I work with tell me it doesn't really blow the cell up like that. Now, so one of the things we've done, and that led to a lot of the products I showed you a couple slides earlier, is start companies. I found that when I would try to get large companies interested, they wouldn't do it. They might do it for a little while, but they'd just give up. So I got involved in starting companies, and I kept doing that. And I'll just tell you about one of them. In 2010, myself, Nubar Fayan, who's on the MIT Corporation, in charge of flagship ventures for pioneering. Karen Chen is a cardiologist, and Derek Rossi. We talked about starting a company to make messenger RNA therapeutics. This is, and I had consulted for Genentech for over 30 years, and I've watched the great work that they've done on making protein therapeutics. But making protein therapeutics isn't so simple. You have, even though probably 70% of the top selling drugs today, of the top 20 are proteins, if you try to make a protein therapeutic, you have to have these giant reactors, or let's say in the case of vaccines, like flu, you take eggs, and it takes a year to grow up the stuff, and that means you have to guess at what the vaccine should be. Should it be H1N1, H1N2? That's why the flu vaccines are like 40% effective, because you're guessing. But with messenger RNA, if you could put it in a nanoparticle, like I just showed you, and protect it and inject it in the body, well, the body will do all the work. It will make the protein. You don't need any giant factory or any eggs. So that was the idea. Not only that, you could possibly, with messenger RNA, make proteins from it that treat diseases that could never be treated before, because you could have intracellular membrane-bound proteins. So I should say, if you go back and look at what happened with Moderna starting in 2010, lots of scientists criticized it, said it would never work. Every pharmaceutical company pretty much ridiculed it. And almost every stock analyst and newspaper ridiculed it. Like Stat, which is one of these things, they kept saying, well, it's valued at 2 or 3 billion. It's ridiculous. It's so far overvalued. At any rate, so there was all this criticism. Nonetheless, the people at Moderna, a lot of whom are MIT graduates, they did fantastic work. And by the end of 2019, we had 13 products in early stage clinical trials. And then COVID hit. And I want to show you what the Moderna scientists did, which I thought was pretty remarkable. So, on January 11th, the Chinese scientists published the virus genetic sequence for what's called the spike protein. It took less than 2 days for the Moderna scientists to design and finalize the vaccine, and then put it in nanoparticles. Our clinical partner was the NIH, and they could begin the preclinical testing. And by March 16th, just a little over 2 months after the start of this, the first patients were injected in Seattle. What you do whenever you're doing a clinical trial is the first trial is what's called the phase 1 trial. That's looking at the highest safe dose. So that's what we did, and within another 2 months, we had the initial data. And I think everybody at Moderna, myself included, thought it was pretty good. We have to announce it because you're a public company. And when it showed it was safe and there were antibodies. And I would have thought that the press and the stock analysts and the physicians and the newspapers would have thought this is a good thing and want to be happy. Let me just show you the headlines of our own local newspaper on May 19th, 2020. So the headline, front page headline, is "This is not how you do science." Criticizing Moderna. Of course, my picture is underneath it. So that was, well, anyhow, just to continue the story. When you, I mean, a lot of you know the story, but I'll just tell you my personal. So this was phase 1 trial. Then you do a phase 2 trial to look at safety, to look at efficacy. And then the key trial is always a phase 3 trial. A phase 3 trial is, in this case, 30,000 patients. 15,000 get the treated one, 15,000 get what's called the control, placebo control. And it's double blind. Nobody knows who got what at many hospitals. So 6 months later, after this article came out, I'm sitting in the boardroom. I was on the board of Moderna from the beginning. I'm still on the board. But this is now November of 2020, and they broke the code. And the results, and this was in all the news this time, were striking. Basically, as you may know, not a single of those 15,000 people died. Not a single one of those 15,000 people even went to the hospital, and it was 94% effective. Flu, for example, is 40% effective. Just to show you something. Thank you, but there's so many people at Moderna who deserve the credit for it. This is the picture of the vaccine, and I'll just show you some data. So basically, this is just looking at hospitalizations. You can see the enormous difference from the CDC between unvaccinated, vaccinated without a booster, and now vaccinated with a booster. And if you look at the different vaccines, I hope people realize how hard this is to do. You know, so the Moderna vaccine was 94% effective. The only other mRNA vaccine, and the only other vaccine that's yet to be approved, that's still approved by the FDA, there's only 2. There, over 50 vaccines were produced, you know, and went into clinical testing. Only 2. And both of them, messenger RNA and nanoparticles, both of them, tiny little companies, Moderna and BioNTech. I mean, Pfizer came in later, which was great because they were able to help BioNTech, but only those 2 are approved. 50 other ones, as Forbes put, are failures. If you look at COVID drugs, 200 are failures. 1 got approved by FDA. So it's been a very hard thing to do. And the Johnson & Johnson one that got pulled off, that's a DNA, that's an adenoviral vaccine, but that's certainly not bad, but it never got full approval. If you look at the New England Journal of Medicine, here what you can look at is effectiveness of these 2 vaccines with almost 400,000 patients, and you can see that with the Moderna vaccine, mRNA 1273, it's very effective, you know, very, very low incidence. And if you do another one from the New England Journal, here is over 400,000 US veterans, and you can see the incredibly low incidence of hospitalization less than 0.1%, even after 6 months, pretty much. So it's been very effective, and to me, it's the tip of the iceberg over what you can do by combining messenger RNA with nanotechnologies. Let me give you one even more recent example to illustrate that, and that's cancer. One of the things that Moderna's been working on are what are called personalized cancer vaccines. And that is that you could actually take a patient's tumor and actually see if, compare it to regular cells and see how many mutations you can find. You could take the top 34 and take their genetic code, those are called neoepitopes, and make it into a messenger RNA vaccine in a pretty much identical way that you did for the COVID vaccine. You make the messenger RNA and you put it in nanoparticles. And the clinical trials that have been done, and these have been announced also, our clinical partners, Merck. Merck has, as you may know, a very successful immunotherapy drug called Keytruda. And what's done is take half the patients and give them that, and the other half, Keytruda plus the personalized cancer vaccine. Each, by the way, patient gets a different personalized cancer vaccine, or, because they have different tumors. So anyhow, the results of this, what's called phase 2 trial, you see a risk of recurrence or death by 44% that's highly statistically significant. And of course, right now, Moderna has 64 other clinical trials going on. And we'll see what happens over time, but I think it really could be a revolution in terms of therapeutics, partly based on biology, partly based on nanotechnology. I want to also now go back in time and tell you some other things we're doing at MIT, which will, I hope, someday have a bearing on these kinds of things. So I was visited in 2012 by a very successful software entrepreneur, and he and his wife have a foundation, and that is aimed at trying to help people in the developing world. So they've been a very substantial funder of our lab, but the way it started was, he said, well, Bob, people in Africa, they don't come back for a second injection. In fact, something like 500,000 babies die every year because of getting tetanus and many other problems. They just don't come back, they don't have the same. So he said, could you ever come up with a way to give a vaccine that would be what we call self-boosting? In other words, you give one injection and yet it keeps coming out. And if you look on the left, on the right, what you see is what we wanted to do is get these pulses, have some come out at time 0, some at a month, nothing in between, some at 2 months, and so forth. So you get these boosts. How could you do that? Well, what we thought is, let's take a particle. Here's the vaccine, or say it's the red. We'll cover it with a material, like a polymer. And that polymer that we chose is actually the same materials in the suture, polylactic glycolic acid. But what we realized is by changing the molecular weights and composition of that material, which, by the way, degrades into water and carbon dioxide, you could actually make those little boxes break at almost any time you want, from like a week, a month, maybe even a year. So that was the idea. And so we came up with a way to do that, to make these. We tried different kinds of printing methods, but none of them worked, so we invented our own. We published this in Science, and we patented it. Bill's actually an inventor on the patent. But what's really striking is it does what I mentioned, that you could get pulses. This is 11 separate pulses with one injection. And what we're doing is we're actually now taking this further. We've actually, Anna Jacquelenik in our lab has led this, and you can actually someday, whether it's the developing world or even our own world, put these in what are called microneedle patches. They're like little Band-Aids. And the idea, if you ever had a microneedle, it doesn't go in very deep, but deep enough so that you'll get the injection, but not so deep that you feel hardly any pain. And you see a picture of some of these microneedles on your right. And on the left-hand side, you see it's like a little Band-Aid, and they put it on in the middle. And so that's what we're doing, or hoping to do with all kinds of vaccines, not just mRNA, but that you could someday give single-step injections. In fact, just Monday, we published in Nature Biotech a way of creating tiny portable 3D printers that could print these microneedle patches all over the world. They're small enough that you could ship, and we were able to stabilize these vaccines. So that's been one of the things that we've done. I wanted to go over a couple of others. So as I mentioned to you, I worked in the hospital, and one of the things that I noticed when I was working there is that pretty much any time somebody did something in the biomaterials area that was driven by a clinician, not a chemist or an engineer, and what they did is they'd go to their house and find some object that kind of resembles the organ or tissue they wanted to fix. So the artificial heart is actually a lady's girdle, and they picked that because it has a good flex life. Breast implants, one of those is a mattress stuffing because it's squishy. And I thought, well, why can't we use chemical engineering design to design from scratch materials that we could do something with? So in the area of degradable polymers, we picked an example. When we started, only one, the material, the polyesters, were approved by FDA, but they dissolve like you see here. They start out like a phone book and get spongy and fall apart. That's okay in some cases, but you can imagine that if you had a toxic drug like insulin or an anti-cancer drug, it could just dump out and could kill somebody. So we said what you really want is surface erosion, layer by layer erosion, and it couldn't dump. So I won't show you the chemistry, but what we did is we made what we call a copolymer. We had unit A and unit B, and by mixing them together, we could make these materials dissolve at any rate, and if we made them very hydrophobic, water repellent to keep the water out, and yet use certain bonds like the anhydride bond, we could make them dissolve. We could make them dissolve. And here's just a graph of that. Depending on the ratio of those two units, the A and the B, we could make it last from 2 weeks to many years. So once again, I wanted to see if we could treat an important disease. And here, Henry Brehm, a young neurosurgeon, now chief of neurosurgery at Johns Hopkins, wanted to treat this disease. And it's glioblastoma multiforme. It's uniformly fatal. And at this time in the 1980s, when we started this, the only drug that was approved was this one, BCNU. And it's a terrible drug. You give it, it travels throughout the body, causes toxicity in the liver, kidney, and spleen. But what we thought with Henry is maybe we could change the paradigm. Maybe rather than give it systemically, we could give it locally. Allow the neurosurgeon to operate on the patient, because they're going to do that anyhow. But before they close the patient's brain up, they put little wafers in of BCNU with this material. Now normally that drug lasts 12 minutes. But now we can make it last whatever amount of time the neurosurgeon wanted, say a month, which is what he wanted. And it protects the drug from degradation. And because you're giving it locally rather than throughout the whole body, you only expose largely the cells you want to the drug. So we came up with this idea, and of course, if you're a professor, you have to raise money. So we write grants, and we just did terribly. I entitled this next slide, "This approach will not work." When we first did this, the reviewer said the polymers can't be synthesized. But I had a very good graduate student, Howie Rosen. Howie later became president of ALZA, a big $12 billion company, and became elected to National Academy of Engineering, and he showed you could make them. Then we sent it back again, and the reviewer said, well, look, you have these anhydride bonds, they're going to react with whatever drug you put in. So that's never going to be useful. But I had another couple of postdocs, Bob Linhart, who went to RPI, and Cam Leung, who's now a chair professor at Columbia in bioengineering and been elected to the National Academy of Engineering and National Academy of Medicine, and they showed that there was no reaction. Then the reviewer said their polymers are fragile. They're low molecular weight, they'll break. I had another couple of postdocs, Edith Mathiowicz, who's a full professor at Brown, and Avi Dome, who's now dean of pharmacy at Hebrew University, and they showed that you could make very strong materials that wouldn't break. Sent it back again, now 1986, and they said, well, but these are new materials. They've never, ever been tested in people or animals before. They're certainly going to be toxic. I had another graduate student, Cato Lorenzen. Cato later became dean of medicine at University of Connecticut. In fact, President Obama gave him the National Medal of Technology a few years ago. He showed that they were very safe. Send it back again. And the reviewer said, well, okay, but the drug will never diffuse far enough to kill the remaining tumor. Well, I had another graduate student, Mark Krausnitz, who Yale hired as their inaugural professor of biomedical engineering to start their biomedical engineering department, and he's been elected to multiple national academies, and he showed that it did diffuse far enough. This kept going on and on until 1996, when the FDA approved it. It was actually the first time in over 20 years they approved a new treatment for brain cancer. First time they ever approved this idea of local chemotherapy. You can probably tell from the way I'm talking that I'm very proud of how well all the MIT students did. They all got major awards, great positions, and things like that. The reviewers from the other universities, they didn't do that well. I do want to show you what this looks like, but again, if anybody's squeamish, don't look. But here, you see a little wafer going into the brain, several of them, and then you close up the brain. You know, it's very hard for me to get advice, but a number of years ago, I was giving a lecture at a chemical engineering department. My wife came to it, and I said to her at the end, you know, after she listened, I said, what did you think of the lecture? And she said, well, Bob, she said, the lecture was okay. That's actually incredibly high praise. But she said, you know, you had those 2 bloody slides on for 10 minutes and everybody, all those engineers were turning green. So ever after that, I did what I did today. I gave a warning. And so, but I do want to tell you the sequel to that was that a couple of years later, I was asked to give this dinner speech. And it was to a group of neurosurgeons. And I told them that same story. And at the end of it, a bunch of the neurosurgeons came up to me and they said, you know those 2 bloody slides you showed during dinner? I said, yeah. They said, those were fine. They said, no problem. You could have left those on as long as you wanted. They said, but those chemical formulas. So leave those on for a neurosurgeon is really bad. At any rate, what you can see, there's no cure for it, but what you can see is that this is an early study and it would have improved survival. This was a more recent study analyzing 60 clinical trials, and again, it's statistically significant, you know, prolongs life and relieves suffering, but there is still no good way to treat this. But what it would also do is open the door for areas that you wouldn't necessarily anticipate. And even though we did this, we also filed patents on how you could use it for all kinds of things. And I had a very good graduate student once. Now he's head of IMS. This is Elazar Edelman. He's an MD as well as an engineer. And also there's companies like Boston Scientific and Angitech in Canada. And so using these same principles, and actually our patent, what they did, I don't hopefully hear people don't have stents, but if somebody has a heart disease, one of the main ways of treating heart disease is to put a stent in. It's like a Chinese finger puzzle. Well, you may know, if you put a regular, what are called bare metal stent in, often it causes a problem called restenosis. You get smooth muscle cell proliferation, the patient could die at minimum, you'll have to reoperate. It happens about 50% of the time. So now what they do is you take Taxol, another anti-cancer drug, not unlike BCNU in some ways. You take that, but you can, because you coat it onto the stent, you're delivering it locally. So you get away with 1/1000th the dose that you would if you do it clinically. And clinically, if you took 1,000 doses, you kill somebody. But locally, what it does is it works. It prevents the smooth muscle cell proliferation. Millions of these are used every year. I thought I'd end the talk with one other idea, and that is how you could use materials not just to deliver drugs, but cells. So here I've collaborated with Jay Vacanti, who's a surgeon. So he asked me, it must have been over 40 years ago, could we ever make new tissues and organs from scratch? And we had this idea, where you could take different cells. Now you could also add stem cells. This paper's been cited over 9,000 times in Science we wrote it. But basically you could take any cell type, and if you give it the right microenvironment, the right polymer scaffold, the cells can reorganize and make a tissue. And these are just some simulated ones. Just to show you a close-up of the scaffolds, you see materials and cells. The other thing this opens the door to is creating tissues and organs on a chip, which could someday greatly minimize and maybe speed up, but minimize animal and human testing and maybe even speed them up. So Gordana Vanyak, who's now a professor at Columbia, who was one of my fellows, and Milika Ritzik, who's one of my graduate students and professor at Toronto, they created a heart on a chip. And this is just the characteristic cells, but I want to show you a video of the heart on the chip. And we're not shaking this or anything, but literally you have 6 million cells, beating as one. Lisa Freed was also involved in this. And you could even take an EKG of this, and which is shown here. But Gordana and Melissa, they actually created a company to do drug testing with these things, and you could tremendously screen drugs for heart treatments. I thought I'd end on one last slide, which is that you can take these principles and make new tissues and organs on many things. And so we're working on cartilage, spinal cords, hearing loss, GI tracts on a chip, liver. I have a picture of me and Julie Andrews, if you look closely on the top. We're working on creating new vocal cords with Steve Zeitels, that's her voice surgeon. We're working on new blood vessels. Laura Nicholson, who was one of my postdocs, she now is a full professor at Yale, and also started a company called Humacyte, which is in phase 3 trials on blood vessels. Skin, heart, as I mentioned, pancreas, and on the middle bottom, blood vessels. We're working with Li Wei Sai. I have a great postdoc named Alice Stanton, and we're creating brains on a chip with 7 different cell types. So let me stop there. It's been a pleasure to speak to you tonight. I'm happy to try to take some questions if people would like, though you might be hungry, but happy to go over it. Thank you so much for having me.
Questions, put your hand up and I'll bring you the mic.
Given the huge success of the mRNA vaccines, is there any way to speed up the approvals of follow-on mRNA vaccines? It seems like we're back to the old take years and years and years approach.
Yeah, well, I think it's a question of politics, right? I mean, that's the politicians in power and what they're doing. I wish there was. I think it's a mistake to, again, I think they've been shown to be safe, and I wish there was a way to do it. That's, unfortunately, it seems to me it's a political issue more than anything else, unfortunately. I wish it were not. When I was chair of the FDA Science Board, that was many years ago, you know, one of the things that we made a clear point of is that all decisions should be science-based. It's not the world we live in today, as you can see from both parties.
Dr. Langer, Steve Jobs said a famous quote, "You can only connect the dots looking backwards. You have to trust that the dots will connect in the future somehow." Given your prolific career in patents and discoveries, do you agree with Steve Jobs, or what would you add to his view of that?
I think, well, actually what he said, I think is largely right, but I think sometimes you can also figure out things that, you know, will have an impact. But certainly, you know, I certainly would not have realized or dreamed in 1974 to 1976 when we were developing these tiny particles that COVID would come along and that nanotechnology would be important for it. That being said, I mean, you know, when you create what I'll call a platform technology, which that is, and which a number of other things are, and MIT has been good at creating them, I mean, that I think it will find applications. You know, and I think the challenge sometimes is getting the timing right and so forth. But I think certainly, you know, he certainly makes a good point. I think there can be certain exceptions to that point.
Dr. Langer, thank you for sharing all of the times that your groundbreaking work has been rejected with us. I found that part particularly inspiring personally. How do you think we could change how science and technology evolution is done in, you know, present society, in academia, in industry, so that groundbreaking work like this is not rejected 20 times before it, you know, comes into play and makes a huge difference in our world?
Well, I think, so that's a very good point. Let me make 2 different points. I don't know that it's just limited to science and technology. Was it that Margaret Mead or somebody once said that never let it be said that, I'm trying to remember exactly, she's going to say it much better than I did, that a small group of people can't change things. She said somehow it's never happened any other way. And I think there's some truth to that, and certainly you got to see, really, if you look at the mRNA vaccines, two tiny companies, Moderna and BioNTech, both ridiculed. And no large pharmaceutical company would even touch this stuff. I mean, of course now, they're like sheep, every one of them runs in and does it. But I think that it's very, very hard to get people to change what's called conventional wisdom. Now, could you ever change it? I think there's things that can be done to help. I think it's very hard to change human nature, but how could you help? Well, I think NIH, one of my suggestions is, unfortunately, let's see with the review process, which you could see we didn't always do so well in, is that usually what happens when money's tight, people sit around the room and they say, this won't work because. And boy, when money's tight, do they pick trivial reasons. I think you could try to instruct them to say, rather than that, what if it did work? What could be the benefits? And penalize them for not doing that. But it would take leadership. So I think there are certain things that could be done. I suppose also maybe there's other types of things, tax incentives and stuff like that could be given, but it's not easy to change human nature.
Well, amazing talk. My conclusion is I should do a postdoc with you. No, seriously, I have a question. You mentioned a lot of growing organs on the chips. So my question is linked with our knowledge about the development of the organs. I have the impression that we haven't mapped entirely all the mRNA, like gene expression that occurs throughout the embryonic or fetal development. So do you see that happening in the future, given the current limitations of our access to tools to actually map these processes?
Yeah, well, I don't want to say like I'm an expert on what you just asked. I'm not, but I think that the whole area of embryology is really very, very valuable and to understand with respect to these issues. And I also think that people are coming up with better and better ways of doing genomic analysis, proteomic analysis, transcriptomic analysis. So I do feel that more can be done and more will be done, but I don't think we're there yet. I think your points are good.
Two more questions.
Sure.
So I have heard that this has been called the century of biology, and it's very exciting. I'm somebody who loves biology. I come from a background of synthetic biology, and I look at your work in combining nanotechnology and medicine, and I'm very inspired. When you really get down to it, it's all these fundamental building blocks, right, that are very, very interrelated. And I wanted to hear about your vision, and this is kind of a broad question, but I wanted to hear about your vision of sort of where the line between biology and nanotechnology and medicine will begin to blur in the coming decades. I want to hear about what you think about how synthetic biology and nanotechnology will look like in the next 10, 20, 30 years, and whether they will sort of converge.
Well, I mean, it's a great question, and of course there's many dimensions to that question. I think in a way they have kind of converged in the mRNA systems. Synthetic biology can be used to make different components that can be used in mRNA technology. And I think there can be more things like that. I mean, what I see is synthetic biology may offer, new, ways of putting, for example, switches into cells that could, maybe make them do things they couldn't have done before. I mean, for example, Tim Liu, who was one of my former postdocs, and he worked with Jim Collins, and Jim, they've started a company called Senti, which is working on synthetic biology therapies, where you could have switches that could maybe make cells more active when they're near a tumor or things like that. I think there are probably many, many ways that people could be creative and combine the things that you were talking about. And I think, different professors at MIT are doing things that are certainly relevant. I don't know that our lab per se is combining the two so much, but there are places like Jim, and Tim was a professor there too, and there are other, Gio Traverso has been involved. So I think there's a number of places that you'll see that.
We have time for one last question.
As an MIT alum working in healthcare, I want to add just one last perspective. You have tremendous accolades, but you have achieved what many of us consider the highest calling that a human can achieve. You have saved many, many people's lives.
I was just going to say, that's a very, I'm happy to do another question, but I was just going to say, that's very kind of you, but really, there are so many people involved in it. I mean, Moderna now has, you know, something like close to 5,000 people, and BioNTech, you know, and there's so many people that really contributed. You know, I played a small role a long time ago, and maybe a role again in helping get some of this started, but it's a team effort. Thank you very much.
So, thank you for a great talk, and thank you for everything you've done for mankind. As you know, Moderna spent 10 years, I think. Before that, one would think that mRNA can be used to treat an unlimited number of diseases. And yet, Moderna spent 10 years, I think, with not getting any approved drugs. And then COVID came along, and now Moderna has 30, I think you said 30 or 40 drugs in clinical trials. What happened?
I mean, in terms of why could you do it so quickly?
Yeah, no, why did it take 10 years and then all of a sudden this
well, I think there's
technology has flourished?
Yeah, well, I think really the reason comes down to politics and money, you know, I mean, and the FDA issues and so forth, and the crisis. So my feeling is, I think, you know, sometimes people think 10 years is a long time. I had some idiots in the New York Times call me up and say, Moderna hasn't, you know, how could they compete with Merck, you know, and other things like that, you know, and they don't even have a drug out yet, and they never probably will. And I said, you know, and I gave the angiogenesis example, that took 28 years, you know, from the start of our science paper before Genentech did it. Well, one of the reasons Genentech did it is they spent billions and billions of dollars. I mean, Moderna didn't have billions of dollars.
Now it does.
Yeah, well, now it does. but that is part of the reason is money, and because of really the people that started Operation Warp Speed. I mean, that was a huge deal, both in terms of the amount of money it gave the Modernas of the world, and in terms of accelerating the FDA. You know, and of course people were dying right and left, and so, you know, that urgency, I think, also made a big difference. But I don't want to oversimplify it. I also think it's complicated. The reason why the FDA doesn't move quickly, it goes back to this Hippocratic Oath about first do no harm. People are very concerned, and sadly, one mistake, you know, and certainly that happened with the thalidomide, one mistake in terms of an approval can just shatter public confidence, you know, in these things. Thalidomide, you know, caused all these birth defects. But so I think the regulatory authorities go out of their way to take a lot of time so that those things don't happen. And I don't want to say there's a simple answer to this. I'm not trying to even take a side. I'd love to see it happen faster. But I think you have to recognize that if it happens faster, you know, some bad things may happen too. And I just think, and then the question is, and this is where the politics comes in, you have to balance the one versus the other. And that's where leadership comes in, and it's unfortunately, it's not necessarily science leadership.
How did you manage not to get discouraged in the early days?
I was discouraged.
But you didn't stop.
What's that?
You didn't stop.
No, but I was depressed, and you know, I was lucky. I had some good friends that I could talk to and were helpful to me, and but I was depressed. I don't stop easily, but you know, there was a time when I was, you know, I was pretty discouraged. But that, you know, that's part of life. Yeah. I've often said that the people that I think are very successful are the ones that in science are often ones that know how to deal with failure well, because certainly for me, I failed a lot more than I've succeeded. But if you don't, but it's easy to deal with success, anybody can do that. But dealing with failure, that's a much harder thing. And it happens a lot, especially if you try to do things like I've talked about tonight.
I think that's a great note to end on. Yeah, definitely